Okay, good afternoon, and today I really like to focus on nuclear energy. And I know that nuclear energy has the power to really polarize people's opinions, but I hope that in the next couple of minutes I'll be able to convince you that we in Miguel's lab have discovered a novel way in which nucleus itself can generate ATP and energy independent of the mitochondria. So to do that, you need to understand a little bit about our model system that we use. And in the lab, we use progesterone stimulation of breast cancer cells. Now, what happens is when we treat these breast cancer cells, they're T47D, with a progesterone analog called R5020. What you see within minutes, a very rapid activation of the kinase signaling cascades. This results in the recruitment to chromatin, where we have the recruitment of chromatin modifiers and histone modifying enzymes. Within hours, we still have the activation of kinase cascades, in addition to the activation or repression of around 3,000 genes. After days of treatment, you find that these cells will actually proliferate, and we've done a lot of work in the lab, very nice work, describing the role of TADs and chromatin modification in this process. But what I'd really like to focus on is the role of poly-ADP-ribose. Now, we're all very aware of post-translational modifications in so much as phosphorylation, methylation, acetylation, etc. However, what I'm really interested in is in poly-ADP-ribosylation. Now, this modification is carried out by a group of enzymes called PARPs, poly-ADP-ribose polymerases, and these PARP enzymes catalyze the addition of ADP-ribose units onto their target proteins. But the amazing thing about poly-ADP-ribosylation is that you don't just have one unit. You have the addition of sequential units, and this chain can be up to 200 in length. In addition to that, every 30 or 40 units or so, you have a branch point when you can form a new chain. So if you imagine the structural changes this causes to the target protein, in addition to being super highly negatively charged, there's a reason why poly-ADP-ribose was called the third nucleic acid. As you can imagine, the functions of PAR are quite diverse. So initially it was shown that when you have PARylation of histone H1, for example, you would have a decondensation of the chromatin. When you have the modification of target proteins by PARylation, you can either increase or decrease the interaction between other proteins. You can activate or deactivate enzyme activity. But also what's very interesting is that the free PAR itself, so the chain itself, has the ability to recruit histones. Indeed, histone H1 has a higher affinity for the PAR chain than it does for DNA. So this work I'll present today all came from an observation, in 2012, where we saw that when we treated our cells with progesterone, we saw a rapid accumulation of poly-ADP-ribose. And you can see here, this is the staining for poly-ADP-ribose. After 15 minutes within the nucleus, you have a huge accumulation. But what really got our attention was that this is a transient response. So if you look here in black, this is the levels of PAR in cells over time with hormone treatment. And you can see this peak around 15 minutes, 30 minutes, that decreases. So this got us thinking to, why would you generate poly-ADP-ribose only to degrade it afterwards? It really got us thinking in the energy required to form poly-ADP-ribose itself is very expensive, and it makes no sense just to produce it to degrade it after a couple of minutes. So going back to energy, if we think about the nucleus itself, the energy requirements of the nucleus are immense. The amount of energy it takes to remodel chromatin, for the activation of kinases, ATP-dependent remodelers, is huge. And it's been postulated that to remodel a single nucleosome requires around 150 molecules of ATP. So we thought, well, the demand of the nucleus is always met by the mitochondria. ATP diffuses freely from the mitochondria into the nucleus. But a key point is that ATP cannot be stored in any way. So what happens when you have an increase in demand? When the demand of the nucleus is not met by the mitochondria, where does the energy requirements come from? And indeed, you'll see in cells that have a high increase in poly-ADP-ribose, if this increase is not stopped, the cells will actually die. So the energetic requirements really got us thinking. And we came up with this very simple idea. Could ADP-ribose be a source of nuclear energy? Could we be generating poly-ADP-ribose only to generate ADP-ribose single units? Could ADP-ribose be a source of ATP specifically in the nucleus? And indeed, this would be independent. What if we could generate ATP in the nucleus independent of mitochondria? We were not the first to suggest this. Indeed, in 1957, Vincent Allfray suggested there was some complex, and energy was contained within this complex to provide a source of energy in the cells. Now, to investigate this, we had to measure ATP, obviously, specifically within the nucleus. So we utilized the A-team, which are fret-based constructs, where when ATP is bound, you have an emission of YFP, and when it's unbound, you have an emission of CFP. Therefore, looking at the ratio between YFP and CFP, we can actually say how much ATP is present. And these constructs are targeted to the mitochondria, the nucleus, or the cytoplasm. In today's talk, I'm only really going to focus on the nuclear construct. So this is a construct specifically targeted to the nucleus, and then we can measure live on the microscope. We treat ourselves with hormone. Every 1 minute, we take the ratio between these two, and this gives us the ATP levels and how they fluctuate in response to hormone. So you can see here is an example of what happens when we treat our cells with hormone live on the microscope. So here you have a snapshot every 5 minutes, and you can see in red this is an increase in ATP, and you can see on the video here around 35 to 40 minutes the ATP levels in the nucleus specifically increase. This is the quantification of a large number of cells, and you can see that when we treat our cells with hormone, we have this rapid increase in ATP. But why was I talking about poly-ADP-ribose if I'm talking about ATP? So then, in order to investigate the role of poly-ADP-ribose and ADP-ribose in ATP formation, we had to have a method that was more flexible and more user-friendly than the A-teams. And to do that, we used the luciferase-based constructs. So again, they're targeted to different cell compartments. And this is ATP using luciferase. And we can see again, at 30 minutes to 45 minutes, an increase in ATP, which is blocked when we inhibit the formation of poly-ADP-ribose or the degradation of the poly-ADP-ribose chain. But that only leaves us with ADP-ribose. We still have to get to ATP from ADP-ribose. So we performed an IP against all the PAR-related proteins in the cell after hormone, and we found this very interesting protein called NUDIXS5, which catalyzes the hydrolysis of ADP-ribose to AMP and ribose 5-phosphate. So does NUDIXS5 generate ATP from ADP-ribose? And indeed it does. If we look at the levels of ATP in response to hormone in control or NUDIXS5 siRNA, we see that ATP is no longer generated specifically in the nucleus when we don't have NUDIXS5. But everybody can speculate that this is an indirect mechanism of measuring ATP by the A-teams or by luminescence. So we actually generated recombinant NUDIXS5, incubated in a tube with ADP-ribose and the presence of pyrophosphate, and we can measure ATP by luciferase, but directly by mass spec, we see ATP in the presence of pyrophosphate, and also by NMR, proving directly that NUDIXS5 can generate ATP from ADP-ribose. And this has a role in progesterone-regulated gene expression, in so much that genes are not regulated when you don't have NUDIX, PARG, or PARP, and that chromatin remodeling and the displacement of histone H1 and histone H2A are abrogated when you don't have the function of these three enzymes. Showing that we can generate ATP locally in the nucleus, independent of the mitochondria, with the big point being that we can now store energy in the form of ADP-ribose. ADP-ribose is formed with specific locations within the nucleus, and now the remodeling machinery and everything else that requires ATP has a source of ATP directly where they need it and when they need it. And of course, we'd like to investigate the implications of this as far as nuclear organization, cancer, and DNA damage. I'd just like to thank Miguel for giving me the opportunity to work in his lab. The ATP measurements by mass spec and NMR were performed by Llanes and Antonio Vaneda in Valencia and Reus. And thank you very much for your attention. I'll take any questions.
Thanks a lot.